Today's question is, which one of the following is not involved in the secretion of potassium from blood into the tubular fluid by the principal cells of distal tubules? First, let's understand the process that has been asked in this question. Potassium is secreted from the blood into the tubular fluid via principal cells in the late distal tubule and collecting duct. This is achieved by active uptake of potassium from the blood into the cell by the basolateral sodium-potassium ATPase pump. This is followed by passive diffusion into the tubular fluid through apical channels. Now, let's look at the options. The first option is big potassium channels or BK. This choice is incorrect because BK channels are involved in this process. These high conductance or maxi K channels sit on the apical membrane of principal cells and are specifically activated by increased tubular flow rates to rapidly secrete excess potassium into the urine. The second option is renal outer medullary potassium channels, which are abbreviated as ROMK or ROMC. Again, this is an incorrect choice because these channels are also involved in this process. ROMC acts as the main pathway for constitutive baseline passive leakage of potassium out of the principal cell and into the tubular lumen along its electrochemical gradient. Let's move on to the next option, that is potassium chlorine cotransporter or KCC1. Again, this is an incorrect choice because KCC1 is involved in this process. This electroneutral cotransporter is functionally present within the distal tubule cells to assist with potassium excretion and manage internal chloride concentrations, ensuring ionic balance is preserved during high transport workloads. The next option is 1 sodium, 1 potassium, 2 chlorine symporter or NKCC2. Now, NKCC2 is not found in the principal cells of the distal tubules or collecting ducts. Instead, it is highly expressed in the apical membrane of the thick ascending limb of the loop of Henle, where its primary physiological role is the reabsorption of sodium, potassium and chloride from the tubular fluid back into the cell rather than potassium secretion. So, the correct option is 1 sodium, 1 potassium, 2 chloride symporter or NKCC2. Do visit my channel Simply Biology with Manisha for more such videos. Thank you.